include in our clinical trial sites. So subtopic 1A, this is focused on facilitating and enabling a highly functional and compliant clinical trial investigator network. And there are 4 work packages, and I'll just very briefly go through these. The first of which is all about communication, so communication within the members in the topic 1 investigators, and then more broadly with all the partners as in the IMI proposal. Topic 2 is to provide training to groups that perhaps haven't run clinical trials on antibacterials in the past but would want to do that. So there's training opportunities to create compliant sites. Again, ideally, some of this we would like to focus it in areas of perhaps high resistance that we haven't been running clinical trials in those regions previously. The third point is the third work package is around surveillance, and there's a number of different objectives with that. The first one is when we create a new antibiotic, we want to understand whether there's any pre-existing resistance to that molecule. So we want to evaluate large numbers of clinical isolates in different countries and different regions around Europe. So, for example, we might find pre-existing resistance in one country, and that might adapt how we would use the antibiotic in that particular country. So that's one aspect of what we're trying to do there. When we do those surveillance studies, we'll also be running currently available antibiotics as control, so that will also give us some insight around resistance trends to antibiotics that are currently available, and would like that to be sort of harnessed into other surveillance programs that are already ongoing around Europe. And then the last point is to gear this to perhaps identify specific regions and specific institutions that have high levels of resistance, for the reasons I pointed out previously. And the last point, again, plays to the theme. Our clinical trials at the moment are very large, they're very complex, and they're very expensive. If there is, this will be exploring new designs, such as Bayesian designs, to see if these approaches could be implemented as a more efficient way of evaluating our molecules. So topic 1B is focused on evaluating the safety, pharmacology, and the efficacy of GSK322. So this is a novel mechanism antibiotic. It covers the key causative pathogens of community-acquired pneumonia and MRSA, and is to be available in both oral and IV formulations. So currently, there is no drug that exists with that spectrum and that combination of attributes. So it really has a unique spectrum and formulations. The other aspect of this trial is to evaluate novel rapid point-of-care diagnostics in the clinical trial to see if these tests could actually be used to enrich our clinical trials with the right type of patient. And maybe this could actually be used as a springboard to create new technologies, to create diagnostics that could be used in the clinical setting to enable our new drugs to be pinpointed and used against the right patient in the future. So with respect to the work packages, there's basically one clinical trial to evaluate this molecule against significant and severe skin infections, and a second trial to evaluate the molecule against moderate and severe community-acquired pneumonia that requires hospitalization. If those 2 trials are successful, then that would trigger the progression to the subsequent trials that would lead to the registration of the drug and making the molecule available to patients. Just a few slides to give you some background on this, and we can talk a lot more about this if you would like. So the mechanism of this molecule is through inhibiting an enzyme called peptide deformylase. This enzyme, the role of this enzyme is to remove the formyl group on the terminal methionine on the amino acid as it's growing out of the ribosome. If that formyl group does not get removed, that's a lethal event to the cell, and that's the mechanism of how the drug works. So there's no cross-resistance to any of the currently available antibiotics to this drug. We've successfully completed a phase 1 study and successfully completed a phase 2 study against significant skin infections, which were largely caused by MRSA. And just to give you an overview of the profile of the drug, as I said, covers the key causative pathogens of community-acquired pneumonia, both atypical and typical pathogens, as well as MRSA with the oral and IV formulations as well, which gives clinicians new options in how to tackle these types of infections. So topic 1C, there's a caveat to topic 1C at this point. We are not asking people to apply for this call. This is something that's going to, should be announced later on in this year if everything goes well. So this is really just to give you a heads up of a future topic that will come hopefully out later in the year. The two key objectives of this is firstly to understand the epidemiology of surgical site infections across hospitals in Europe. The second objective is to evaluate two antibodies, one to study its ability to prevent MRSA infections, and the other for the treatment of sepsis. So the two work packages here, work package 6 is focused on the Staph aureus monoclonal antibody. The first work package there is to get a better understanding of the epidemiology of surgical site infections across Europe. The second component there is to understand the role of virulence factors in causing these surgical site infections. And then the final two work packages is to actually evaluate the activity of the molecule, the antibody, in preventing Staph aureus MRSA infections in ventilator-associated pneumonia, which is associated with a high level of mortality, and surgical site infections. And then work package 2 is focused on evaluating this antibody against severe sepsis and including septic shock. I'm sure many of you are aware that sepsis is a huge unmet medical need in Europe as it is around the world. So this will be the subject of a future call. So the expected contributions from applicants and the expertise and areas of interest that we're looking for. Obviously, we're looking to hear from experts in serious hospitalized bacterial infections. These molecules I don't talk about, the focus of their use will be in the hospital for significant infections that require hospitalization. We're interested to hear from hospitals and healthcare institutions that are interested in running the phase 1, phase 2, or phase 3 clinical trials that are in the call. There's opportunities for small, medium enterprises to provide training to create compliant clinical trial sites, perhaps in areas that we haven't been before, which have high levels of resistance. There's also opportunities for groups with rapid point-of-care diagnostics that are ready to be tested in a clinical trial setting. Also, clinical research organization with experience of running clinical trials across Europe and globally. And also those with expertise in statistics, PK/PD, and also with experience in conducting and running the clinical microbiology surveillance programs that I touched on in one of the work packages. The contributions from FPIA, well, we'll be obviously providing clinical trial expertise and all the information and what's needed to run the clinical trials on the molecules that I've spoken to. We will also be providing the non-European part of the clinical trial. More often than not, when we run a clinical trial, we have to run it globally. That's the only way we can get a reasonable number of patients on a reasonable timeline to actually prove that the molecule is working. So that's really why we have to do these trials globally, so the FPIA members will be providing the non-European component of the clinical trial. Also, we'll be obviously providing expertise in project and alliance management, statistics, PK/PD modeling, but also knowledge and expertise around these clinical microbiology surveillance programs, and as well as what can be fairly complex clinical microbiology procedures and protocols that are needed in the clinical trials. So that concludes the brief outline of topic 1. Topic 2, remember there's two components to this. I'm just going to briefly talk about the first. This is where we're creating the database of learnings from our successes and failures. So this will be a sort of repository of where we keep all that information. I'm not going to talk much more about that because it's fairly straightforward. What I am going to focus on is the second component of this topic 2. And this really tackles one of the major challenges that we find in the industry. It's quite easy for us, well, nothing's that easy, but it's not a big challenge for us to design molecules that would be very potent inhibitors of novel gram-negative antibacterial targets. That's not a major challenge for us. We can do that, and they can be selective and very potent. The challenge we face is actually designing those molecules so they actually get in to the gram-negative and can get to their lethal target to have their lethal effect. So we see from the industry, this is probably one of the biggest barrier to us creating a robust pipeline of new molecules for addressing multi-resistant gram-negative infections. So the key goals here What we're trying to do here is to create a very vibrant partnership, probably mainly with European academia, but other groups are certainly can be involved, and maybe encourage groups that haven't previously worked in microbiology or antibiotics, that there may be good translational learnings from mammalian systems here that the microbiology world perhaps never really got caught up with. And all of this is to see if we can identify more rational ways of designing molecules to penetrate gram-negative. So how do you avoid bacterial efflux? How can you optimally design molecules so they penetrate porins and gram-negative membranes? Can we actually use the active uptake pathways that already exist in bacteria to actually deliver these antibiotics directly to the target? So we can sort of use a Trojan horse approach, which has been used in the past, but we think there's much more to do there.